Hi, let's talk about how we use QuaC pipeline at our center to help with consistent and standardized QC on genome exome sequencing data. So why perform QC? So QC helps you to make sure that the data you have is of good quality and you could reliably use the data for the analysis that you have planned, and therefore you can properly trust the results that you have derived from that data. So it essentially makes sure that you can trust your data and not waste your time on time as well as resources on your bad data. So there are multiple steps from sample collection all the way to data processed into VCF format from for genome and exome sequencing. So things could go wrong at multiple steps. For example, the sample in and itself could have been of bad quality, or there may have been a sample swap, or there may have been a sample contamination. It could have been from another human individual, or it could have been from other species, or the sequencing data quality could have been poor. In addition, there may have been batch effects that may have been seen as well. So there are multiple QC tools out there to help with performing QC on several things we just talked about. However, executing those QC tools and then interpreting all those QC results and then performing such a QC in a consistent manner and communicating the results after QC to the downstream users. All these things tend to be rather hard. And to make this more consistent and standardized, we have developed a pipeline called as QuaC at our center. So this QuaC pipeline, it runs several QC tools, and then it also assists with interpretation of QC results. So this QuaC was developed using SnakeMake and pipeline. SnakeMake and Python, and this pipeline runs all the jobs in a singularity plus conda environment in order to promote reproducible data analysis. And currently, we use the pipeline at production level, and currently it's at version 1.1. And we supply input. Input to the pipeline is a list of samples in a pedigree format. Each list is considered a single project at our center. So QuaC runs several QC tools. For example, all the QC tools are listed here. They use BAM or VCF files as input. In addition, there are tools that QuaC doesn't run certain tools, but it is capable of consuming their output. In our case, at our center, we run certain QC as part of a small variant caller pipeline, but QuaC is capable of consuming their results. And after running the several QC tools, what QuaC then does is it aggregates all the QC output from multiple tools in a multi-QC HTML report. And it does this both at the sample level as well as project level. So if you are interested in looking at just QC results for the sample, you could just look at the single sample level. Or if you are interested in looking at all the samples in a project, you could look at them in a single HTML file, and this makes it easier to perform QC. And after aggregation, what QuaC does is it includes a tool called as QuaC Watch. So QuaC Watch, QuaC helps with summarizing the QC metrics based on predefined thresholds. So it essentially looks at a QC metric and then compares it to the expected threshold, and it tells you if the particular QC metric passed or failed the threshold, and then it aggregates all these summarized results, and an analyst or the QC analyst can use this output to say, hey, if the sample has passed or not, and if they have not passed, why it may have failed. For example, here is the QuaC report that's available as part of the multi-QC report, internally what we call as QuaC report. So this tells you if a particular metric and a particular QC tool has passed or passed the threshold or not. And then if they have, and if you choose to, you could look at a particular value for the particular metric, and then see why they may have failed. So what happens after QuaC report is produced by QuaC? Somebody still has to manually review them and then log the results. So performing this QC review in a standardized process can be challenging, and to help with this, we have devised a sample QC database, and these are the fields that we use in sample QC database, and the value for this allowed value for this field are controlled using these particular flags, for example, pass, fail, acceptable, poor, for example. Or in some cases, we also have a free text section where the QC reviewer can log the results of QC review. So to summarize, QuaC was developed using SnakeMake and Python, and it runs several QC tools and aggregates the output, and then summarizes the results based on predefined threshold. And it essentially makes it easy to consume the QC results, and we have a sample QC database which is used to log as well as convey QC results to the downstream users. Thank you.